Welcome to The Explainer. I am so incredibly excited to take you through today's topic. When most people think of fungi, they picture a simple mushroom popping up in the yard after a rainstorm, or maybe some mold on a forgotten piece of bread in the back of the fridge. But today, we're going to completely shatter that perception. We're going to look at fungi for what they truly are: absolutely complex, highly evolved biological engineering marvels. So I want to start by posing a bit of a riddle to you. What kingdom of life lacks a mouth yet digests the world? I mean, it sounds literally impossible, doesn't it? But that right there is the fundamental mystery of fungi. They don't have stomachs, they don't have mouths, and yet they are the ultimate consumers, breaking down organic matter on a massive global scale. So how do they do it? Well, it's all about their incredibly specialized architecture. Okay, here's our structural roadmap for today. First, we'll meet this hidden kingdom. Second, we'll map out the actual architecture and plumbing of a fungus. Third, we'll examine their specialized somatic tools. And finally, we'll unpack the fungal factories, how exactly they reproduce.
Part 1. Meet the hidden kingdom. Defining the indefinable. To really understand how fungi operate, we first have to acknowledge that they are an entirely different form of life. They operate on a completely unique biological blueprint compared to plants or animals.
Now, this definition is packed with some heavy scientific terminology, but let's break it down, because it reveals exactly how they function. A fungus is eukaryotic. That just means its cells have membrane-bound nuclei and complex organelles, just like our own human cells. But here is the kicker. They are achlorophyllous. They have absolutely no chlorophyll, which means no way they cannot photosynthesize and make their own food like plants do. Instead, they are chemoheterotrophic. That's just a fancy way of saying they have to absorb pre-existing organic carbon from their environment to survive and extract energy. So because they can't just sit in the sun and make their own food, they've evolved these incredible biological workarounds. Unlike plants, which use cellulose for structure, fungal cells are surrounded by a rigid, durable armor made of chitin and glucans. And get this, chitin is the exact same tough material found in the exoskeletons of insects. Pretty wild, right? Plus, instead of storing energy as starch like a plant would, fungi store it as glycogen and lipids, which is actually much more like animals do. They are these wonderfully bizarre hybrids of biological traits, uniquely equipped to absolutely conquer their environments. Moving on to part 2, architecture of a fungus, the blueprint and plumbing. Let's see how this microscopic blueprint actually translates into the physical architecture of the organism, and finally solve that riddle of how a creature without a mouth goes about eating. This microscopic reality of a fungal network brilliantly illustrates how they pull this off. What we're talking about here are hyphae. These are tubular, transparent filaments that branch out continuously. This is the literal plumbing of the fungus. A hypha exhibits what's called apical growth, meaning it grows entirely from the very tip. It just relentlessly pushes its way through soil, wood, or even living tissue, releasing powerful digestive enzymes into the environment, and then absorbing that broken-down food directly through its chitin walls. Now, the entire body of the fungus is called the thallus, and fungi basically have two primary life strategies for organizing this body. A eucarpic thallus splits its duties very carefully. One part of the organism is dedicated purely to eating and absorbing nutrients, while a totally separate part handles reproduction. It's highly specialized and very common. On the flip side, a holocarpic thallus, like you'd see in yeast, doesn't bother with that division of labor. It just throws all its energy into a phase of rapid growth, and then the entire body simply converts into a reproductive structure, the whole shebang. Let's zoom back in on those tubular hyphae for a second, because the internal plumbing isn't the same across all species. We have septate hyphae, which have physical crosswalls called septa, dividing the tube into distinct compartments, almost like individual rooms. These septa have tiny pores in them to let cytoplasm flow through selectively. Then we have coidocytic or aseptate hyphae. These lack crosswalls entirely. The whole tube is just one continuous, giant, multinucleate cavity of freely flowing protoplasm. It's an open concept design allowing for super rapid movement of resources. When you take millions of these branching hyphae and weave them all together, you get a mycelium. Think of the mycelium as a massive, living, underground utility grid. It can spread intercellularly across the surface of a host or intracellularly, penetrating right into the host's actual cells. It's just an incredibly efficient, highly evolved network designed for one singular purpose: expansive, relentless resource extraction. Which brings us to part 3, specialized somatic tools, nature's engineering toolkit. A vast network sometimes needs a little extra help to navigate a challenging world, which is exactly why fungi have evolved an astonishing array of structural modifications. Take a look at these root-like structures digging deeply into the surface. Those are called rhizoids. They are short, delicate filaments that act as both crucial anchors and localized nutrient absorbers. You'll find these really commonly in lower fungi, like Zycomycetes, which desperately need stability to push their structures upward. Above the surface, running parallel to the ground, is the stolon. It's a runner hypha that spreads the fungus horizontally across the substrate to rapidly colonize new territory.
And in reality, it looks exactly like this, just tufts of rhizoids aggressively gripping the base of their environment. Actually, what's fascinating is they don't even have nuclei inside these short filaments, but they are absolutely essential. Without them, the fungus literally wouldn't be able to hold its ground against environmental pressures while it sends up the delicate structures it needs to reproduce.
Fungi also have what I like to call their specialized burglar tools. These evolved specifically for breaking and entering. First up, the aprosorium acts as a minute infection peg, generating immense pressure to physically pierce and break through the tough cuticle of a host plant. Once that defensive wall is breached, the fungus deploys a hostorium. This is a specialized branch sent directly inside the living host cell. It balloons out and literally drinks the cell's nutrients from the inside out. And for long-distance travel across hostile terrain, fungi can bundle their hyphae together into thick, tough, armor-plated cords called rhizomorphs, allowing them to safely advance and scout for new food sources. Finally, part 4, the fungal factories, reproduction. Because once a fungus has anchored itself, pierced its host, and absorbed enough nutrients, it turns its full attention to its ultimate goal, propagating the species. Fungi are master propagators, and they use radically different methods to achieve maximum spread. On one hand, we see simple vegetative budding, which is super common in single-celled yeasts. A mother cell simply pinches off a smaller daughter cell, dividing its nucleus and creating an instant clone. But contrast that with sporangia. These are complex, microscopic sacs packed with countless asexual spores, elevated way above the surface and totally ready to be unleashed into the wind. Asexual reproduction is really all about rapid mass propagation without the complicated union of sex cells. A fungus can simply fragment, where a broken piece of mycelium detaches and just grows into a completely new organism. They might use fission, budding, or massive spore production. Some spores, like conidiospores, are born totally free on the exposed tips of special hyphae. Others, like chlamydospores, are thick-walled survival capsules formed right in the middle of a hypha. They're designed to lay dormant and outlast incredibly harsh conditions. But fungi don't just clone themselves. They also utilize highly structured sexual reproduction to ensure genetic diversity. Think of this process as a strict biological assembly line with 3 very distinct phases. Phase 1 is plasmogamy. This is the fusion of 2 protoplasts, bringing 2 compatible nuclei together into the exact same cell. Phase 2 is karyogamy, the actual fusion of those 2 nuclei to form a singular, diploid nucleus. And finally, phase 3 is meiosis. This is the vital reduction division that returns the nucleus back to its haploid state, perfectly primed to form a brand new sexual spore. And what does that biological assembly line produce? Well, it yields incredibly diverse sexual spores. You have oozospores, which come from the unequal fusion of gametes. You have zygospores, characterized by the fusion of 2 perfectly equal sex organs. And then you have the fascinating ascospores. These develop tightly packed inside a tiny cylindrical sac called an ascus. What is truly amazing about these ascomycetes is how they release their spores. They actually build up internal turgor pressure until the tip of the sac explosively breaks down, forcibly ejecting the spores into the air in a microscopic squirt. So we've gone from microscopic, chitin-armored tubes to specialized infection pegs, all the way to explosive reproductive sacs. Which brings me to a final thought for you to ponder. If the ground beneath your feet isn't just inert dirt, but is actually a vast, digesting, highly engineered architectural network of mycelium, continuously reshaping life and death, how does that change the way you see the natural world? The next time you walk through a forest, remember, the real action is happening right under your boots. Thank you so much for joining me for this explainer. Keep asking those big questions, and I'll see you next time.